Okay, hey guys, it's Justin again. We're just going to check on the unknown mixture separation from last week, and I just wanted to show you what resulted from that. So I'm going to go ahead and take the lid off just so we can take a better look at what we got. So from the mixture of two unknown microbes, this is what we have here. Pardon the cracks in the auger here, it just dried out a little bit from it being in the incubator over the weekend. So, but no matter, because we got some good results here. So you could see just from the outset here that we have two different looking colonies here, and we have specifically what I'm interested in is this guy here. It's like a white color, and then right next to it you could see off-white, or I'm going to call it a clear looking colony. So this came out in just the second quadrant, it looks like. So here was my first quadrant and then my second one. The third and fourth quadrants, well, it looks like there's a little in the fourth quadrant here. So but no matter, this is a successful quadrant streak to serve the purpose of diluting out the mixture of cultures so we could see the difference between the two and then isolate them. So how we're going to isolate them today is grab the colonies that are far away from other different looking colonies, because I can tell by looking at it that if I grab this one, for instance, I'm not going to accidentally pick up another one that looks different, and that's going to be good in the isolation process. So what I did is went ahead and labeled two more quadrants, plates, white and clear. So the first one, I'm going to pick up this white colony and streak it out in a quadrant streaking fashion, kind of the same way we did this one, just to make sure that it is indeed by itself. So I'm just going to go ahead, I have sterilized my loop already, and I'm going to pick up this white colony here, and we don't need to scrape it up or anything. So remember, we're dealing with microbes here. A colony of bacteria holds millions of cells of that bacteria, so we don't need to scoop a bunch up. So all I'm going to do is use my sterilized loop here and just touch the top of it, just like that. Replace my lid, go to my plate labeled white, and do a quadrant streak on this. So I'll do my first quadrant there. And just for the sake of time, I'll go back and do the rest of the quadrants. You guys are familiar with that process now. So, but I have the separated white colony on this plate, so I'll go back and quadrant streak that after this is done. Okay, so I'm going to sterilize my loop, and we're going to go for the clear looking colony. So let's take a look. I'm liking the looks of these guys right here, so I'm going to make sure my loop is cool before I touch it. I'm just going to cool it over here where I see no bacteria growing. I know that's not going to contaminate my loop, because you know, if the loop is too hot and you go to pick up a colony like that, you're just going to kill it all, and you're going to waste a lot of time by letting it grow overnight and see nothing grow on it. So it's important to sterilize, but it's also really important to cool it off. Okay, so I'm going to get my other plate ready here that's labeled clear, and I'm going to go for this clear looking colony here that's away from the white. Then I'm going to apply it to the plate labeled clear on my first quadrant. Okay, and I'm gonna let these grow in the incubator overnight at 37 Celsius. I'm gonna go ahead and hang on to this plate though until I'm certain that these other two have grown successfully. Because I don't want to throw this out and then, nothing grew on here and then, that's it for you. You have to get a new mixture made for you. So, but we have a successful quadrant streak separating out our gram-positive and gram-negative microbes, and then I applied it to each of these, which will hopefully successfully isolate them. So on the next video, I'll show you the results of these growing at 37 overnight, and then we'll be moving on to our next steps in the Unknown project. Thank you.